All right, I think hopefully everyone can hear. I'm competing with the rain today, but in Iowa, we also need rain, so I can appreciate that and kind of deal with it. So thank you for the invite. So my group does a lot of work in nutrition and immunology, and over the past 6-7 years, we've also added some welfare and environmental enrichment to our research portfolio. So I think one of the most interesting things about doing research is sometimes when you have an outside view on a problem, maybe you can find ways to solve it a little bit more creatively, just because you're not kind of directly sitting in some of the parts of the field that might be assumptions. So today I'm going to talk to you about some of our welfare work coming from a nutritional immunologist perspective. So please forgive any vocabulary that I might creatively misuse, just because I'm not conventionally trained from a welfare perspective. So every fall I teach a course, and Poultry Science 223, I talk to the students about where poultry production is and where we're going. And I think it's important for us in the industry also as academics or allied to step back and think, where are we right now and where are we going? And it's a good thought exercise because the poultry industry is incredibly flexible, and we respond really quickly to consumer changes. I don't really see that happening in other industries as quickly as we're able to do in the poultry industry. So really over the last 12, 13 years since I've had my PhD, there's been a huge change in how consumers are driving our production. So that's everything from how birds are raised, what they're fed, the marketing that is involved with how we talk to consumers about our products. There's also a lot of interesting changes due to antibiotic-free and the predominance of the market share. So we suddenly have to know how to have health-based or nutrition or welfare-based changes that create a very healthy bird. So I think the last 15 years of research even has really changed how we focus on nutrition, welfare, and housing because of these big global changes. So today I want to talk to you about some really interesting discoveries that were parts of other studies that kind of have a welfare component to them. The first kind of big global theme is some of these immunometabolic changes due to stress that we've been noticing as we apply some new technology to our research with both broilers and laying hens. And then we have a really neat welfare and performance study that's been about half of our work over the last 6 years that has to do with environmental enrichment from a laser basis. So in that study, I'll talk to you about behavior from a laser following perspective, a performance perspective, the effects that we're seeing on woody breast, corticosterone production, and bone density. So something that my group has been doing is trying to develop new ways and approaches to support performance outcomes. There are tons of assays that are kind of been there, done that. Everything from microbiome, colorimetric assays, cytokine assays, PCR, ELISA, et cetera. So if you've read research, you're pretty well versed in these outcomes as things that we measure. There is an interesting problem with reagent availability for birds. So sometimes we're limited in things like flow cytometry just because of the availability. But there are some newer or less common technologies that we've been trying to employ that basically look at some of these changes from a larger view or a more specific view. So the one that I want to talk to you about today is the seahorse metabolic assay. And I'll give you a little primer on what that is and then how we've used it to understand some big changes in the immune system that directly impact bird welfare. So there's a really neat immunological theory. It's called the Warburg effect. You might have heard of it kind of in media when they say sugar feeds tumors. So there's this really neat change that happens in the immune system when the immune system is activated. So the immune cells undergo a specific remodeling of what fuel selection to support their activation. And this change is they're choosing to do glycolysis even if they have other methods of fuel production available. So our hope is that if we're able to measure these metabolic changes, we can apply a challenge to the bird, we can apply a challenge to the environment, or we can apply a challenge basically to the cell itself, and we'll be able to measure their ability to respond to one of these challenges. So we know that manipulating the diet might support immune system function. There's a ton of research that's been done in that area. And our goal is basically to find additional biomarkers, including fuel selection, that might give us insight by using peripheral blood mononuclear cells, or PBMCs, to get information on the inflammatory status of the bird. So when we think about overall stress and immunity, there have been some really cool papers recently that don't provide a direct link, but provide more of a holistic approach to why we care so much about the immune status of the birds. So some of these papers describe kind of on a global level how immunity changes overall welfare. They describe how maybe boosting the immune system for a long period of time can have negative effects on welfare. But we know that there is a direct link basically between how the immune system is functioning and the overall health status of the bird, and then how that bird interacts with its environment. So, for example, we know heat stress causes definite negative welfare concern from many different reasons, but also because it affects the immunity of the bird. We know that light management can change how the bird interacts with the environment, as well as alters the immune system. And there have been some recent compilations of studies that show us that we can use immunoglobulin A, which is a secretory antibody, as a non-invasive physiological biomarker in animal welfare studies. So this is really awesome because this gives us another way to measure how the bird's body is physiologically reacting from an immune status standpoint without having to do an invasive sampling technique. So one of the things that I just kind of want to hit the point home is that from a welfare standpoint, negative or acute versus chronic stressors have different effects on the immune system. And this is coming from a brain immunity axis, which is a very cool new area of research that is also connecting the microbiome. So do we have all the answers yet? Absolutely not. But we know that the affective state of the animal can directly impact how their immune system is functioning. And a great way that we can kind of watch this connection is by studying things like production of IgA in secretory membranes, including the nose, the mouth, and feces. So this is not the be-all, end-all way to measure immune status or immune function, but it gives us insight into how the animal is kind of taking some stressors, including just being separated from conspecifics, or individually housed, or moved to a new environment. So I think this is really exciting to me. It goes past what my group does because we don't work with many neuroendocrine markers, but there are other people at Iowa State and elsewhere that do. So kind of a neat area if this is of interest to you. So the effect of the immune stress, it definitely is linked to growth performance and immune function in livestock. And this is a really cool paper in case you want to delve further, but it goes into some of the specific molecular mechanisms that are linked to stress, such as heat stress or, moving, or some other stressor that could be introduced into the animal's environment. So what the heck is the seahorse metabolic assay? So we know there's this kind of crosstalk between the affective state and how the immune system is functioning. So this assay kind of breaks metabolism down into a simple way, and we either give the cells the ability to use glucose, or we allow them to use the mitochondria and do fatty acid or protein breakdown for metabolism. So just without getting too deep into this, we can use different drug inhibitors to force the cells to use a specific pathway. So then the question after that is, can you still have a specific type of energy metabolism? And if you can, how good are you at it? So those are the kind of the basic questions that we're asking in my lab. And we found differences based on age of the bird, genetic line, and some different stressors that happen. So the thing that we're looking for when we apply a stressor, like I said, is can the bird still have a response? So do the immune cells of the bird still switch a metabolic preference? And if so, how big is that? And then how do our experimental variables change that? So we'll be looking at a particular study that we've done over the past couple of years. So this particular study was done in laying hens, and the seahorse metabolic assay is really interesting because typical labs are using this in cell lines that are identical. So cancer cell lines, basically cells that are all replicates of each other. And my group is doing this in lines that are not identical. So we're using mixed cell populations. So there's some inherent difficulties already with that. But the reason we chose to do this is we can draw blood from a bird and watch a metabolic change over time in response to some different variables. So we took some older laying hens and we had a group that was unvaccinated or a group that got an experimental vaccine. So we produced the vaccine within my group, and so we know it's something that they hadn't had prior experience with. And basically, we're seeing a change in oxidative metabolism due to the vaccine. So this convinced us that in a wild type population, we're able to see that birds do have a metabolic response that we can see in their peripheral blood cells. So we went on to collaborate with Dr. Yuko Sato and Dr. Mo El-Ghazar at Iowa State. They had industry come to them and they had a problem where some laying hens at specific ages were having some high mortality and issues with growth and production during different parts of the production cycle. So there seemed to be a difference between very young hens and very old hens, and they were able to isolate Staph aureus from the birds that came in for necropsy. So we did a study with them where we looked at route of inoculation based on age, and we, in long story short, found some differences in the metabolic immune profile and also the T cell populations that were there. So one thing that we found is that we can see differences in this immune profile based on what cells are around. But I think the most interesting thing that we found was that acclimation periods are critical for the immune response. So within this group, I'm sure there are people that, bring birds in and they do research. When you're talking about day-old chicks, it's kind of interesting because they're coming from a hatcher and they're going into their live environment and they're doing their thing in the research pens or the facility. But it's often the case that when you take older birds, they're already in a housing system and maybe you have to move them to another location for your particular study. So that's what happened in the study here. We moved old and young laying hens from a commercial facility and we put them into our research facility. So iCook says you got to have 3 days of downtime, right? You're probably familiar with that if you have ever written an iCook or done research with birds. But we found actually that the 3-day acclimation period negatively affected the bird's ability to respond to the challenges that we were applying for seahorse. So without telling you some of the specifics of the setup of the assay, I'll just point out that the way we want the immune cells to respond are, oh, my little thing's not working. This right here is the response that we expect at the bottom. So when we apply a challenge, we expect a slope between these two lines to increase and then for it to slowly go down. So if you look at panels A, B, C, D, E, and F, that is not happening in those panels. So what those panels are representing is after 3 days after arrival, we took blood from the hens as a baseline, and then panels C and D are 6 hours after we inoculated them with Staph aureus, and panels E and F are 24 hours after we inoculated them with Staph. So basically, until 3 days post arrival, the birds did not have a normal immune response. And from a welfare perspective, we basically found out, like at a metabolic level, 3 days of acclimation was not enough to give them time to have a normal immune response. So the birds looked cool, right? They were happy. They hadn't lost weight. We've been weighing them. We gave them 3 days to do whatever they wanted to do, find their bird things, get their new neighbors and figure that stuff out. But from a metabolic perspective, if we're doing research, we did not have the proper conditions at the beginning because our baseline measurements told us that even if I applied a drug to force the animal's cells to use the glucose pathway, they just couldn't. So a major outcome of that paper, among other things, was that there is definitely a welfare and an immune benefit to give birds a longer acclimation period before our research is applied. So we had another set of studies in broilers, and part of this work was in collaboration with Dr. Hargis from Arkansas, and we were able to use his necrotic enteritis model. He was amazing and a great help for helping us set up how the model works. So we did a bunch of work to understand what is the difference between giving birds salmonella on day of arrival versus not giving them salmonella on day of arrival and then following the rest of the model. So the model is give the birds salmonella inoculation when they arrive, day 14, give them Emeria maxima, and then follow that up with Clostridium perfringens a couple days later. And basically, we found that there was a huge metabolic difference in the immune cells, whether or not we gave the birds salmonella on day of arrival. So in panel A, we had a different disease burden basically than in panel B. So there's not much change in birds receiving salmonella on day of arrival, along with Emeria maxima on day 14. But in birds that did not receive salmonella on day of arrival, we see a much bigger and different change in their ability to respond to the drug inhibitors. So perhaps there is kind of a difference because salmonella already was around in how the birds immunologically responded later. But we're also kind of wondering from a welfare perspective, those birds that do get salmonella on day of arrival, we can't really tell physically that they have salmonella. They hide that pretty well. But immunologically, we can pick it up that there's a difference. So we're trying to understand what are some of these other changes based on the model to understand long-term, what are some things that we can do to help birds early on, because they're not showing you that they have these sorts of infections, to kind of maximize their first 2 weeks or the starter performance period. We've also seen the same when we give the birds the rest of the treatments. We see that the ability to do glycolysis is different based on which model that they're in. And there's definitely a difference between giving the birds just Emeria only and then salmonella and Emeria maxima. So there's some really, really cool changes that are happening in the bird that are not apparent just kind of like based on visualization. So where are we going now with that sort of project? We're working on the effect of housing system on immune response and trying to connect that to some other welfare parameters. We want to be able to connect stressors to welfare and defects in immunity, which is a very hard task because, there's many aspects to both of those. But if we could, maybe we could have some changes in how birds are, put into new housing systems, acclimation periods, that sort of thing before other things happen to those birds. We're also interested in the systemic effects of immune-related feed additives. So is there something we can give the birds that will improve health and welfare based on immune function early on? And then precision feeding and additive recommendations based on that immune stimulation. So it's a super cool project. We're happy to have found some molecular signatures that help us understand maybe what the bird is hiding because they're not telling you that they've got something going on. So for the rest of the time, I would like to talk to you about our really fun and awesome laser project. So has anyone played with infrared laser thermometers in chicks or just in houses? They love it. They follow and they peck it. This was a video, and I didn't do the video because I've never been successful on Zoom with videos, but I just moved that laser forward and there's 3 birds kind of fighting for that laser because they're trying to play with it pretty hard. So we noticed basically that the chicks were obsessed with the infrared laser thermometer anytime we put it into the pen to check the temperature. So we were interested in using that laser in a larger fashion for chicks to play with during the production period and seeing if there were any behavioral changes. So we've been developing some novel object exploration tests based on enrichment. This is something that's still in progress. We've kind of, over the course of this project, delved into movement and woody breast. We're trying to connect enrichment welfare measurements and behavior outcomes to performance. And we're trying to determine a correlation, if any, between pre-harvest and post-harvest woody breast scores. So those are kind of the big outcomes of where this project has gone. But in the meantime, we've done a lot of really fun stuff, including we've created this platform for birds to walk on. So we can kind of assess lameness over time, but obviously it's called the Kinkwaki Derby. So we have that sticker made. My group likes to have quite a bit of fun. So the studies that we've done involving this laser, we've changed between different lines. We've done Ross 308 broilers and Ross 708, but lately we've focused on the 708 for some of the meat quality aspects. So this is a picture of our initial setup. The lasers are kind of mounted to the ceiling up here. And then we have pens on the ground that are 4x8. And we did the 4x8 pens just to get enough replicates so we could do some behavioral analysis. But the laser plays over 2 of those adjoining pens. As with all things, at some point you get version 2 of your laser. So this is what version 2 is. We work directly with some great people at One Sliding, and I affectionately call this laser the Megatron. It's very clunky compared to this cute little laser over here. But we've basically done the further studies with the One Sliding version of the laser. So initially we have looked at 4-minute laser periods 4 times daily. So we did a bunch of studies in 2020 and 2018 on that. We then got a USDA grant where we're looking at 6-minute and longer periods to see which preference the birds have as far as play, or are they kind of bored with it and do they quit interacting. But the birds are getting the laser enrichment 4 times 6-minute periods, and our next study is going to be 8-minute periods, and we'll just see what happens. So here's an overhead shot of birds towards the end of production in the finisher period, but you can kind of see the lasers playing on the pen, just so it's an overhead view of how those lasers look. So we've published quite a bit on the study as far as the performance and the behavior outcomes and the outcomes that are physiological and behavioral. So if you're interested in those, there's a bunch of papers. But when we originally started this, we were very concerned that what would happen to the broilers is the bird in the middle. Because who knows distance runners that are muscly, right? They usually tend to be kind of, skinnier versus the sprinters, which are, the muscle bound. So our initial work with this laser, we were just trying to keep performance the same and not lose any performance. But it turned out that with our first study in the Ross 308s, we found the birds were more active through week 6 of the study. We also found that the feed conversion was improved incredibly. And so we were thinking, okay, well, they're moving more, but somehow their feed conversion is also improved. So that's awesome, because we thought we would accidentally make birds that were smaller because they were exercising more. We found that their average daily gain was increased, especially in the finisher, which is probably the most important as far as final weight goes. But these all led us to then do further studies with the laser, because our goal was not, we wanted to have welfare improvements, but also we didn't want to have birds that were subsequently smaller as well. So something that we kind of discovered over time was we started moving into the Ross 708 line, and we were wondering if there was any changes to the breast meat quality, mostly because it is kind of a high impact cut on the bird. We saw that there was changes in weight and changes in FCR, and then the next question was where are those weight changes going? So we found that the breasts were larger in different ways, and I'll show you that data in a second. But we also found that the woody breast scores were redistributed in a positive way if the birds had laser exposure. So we had sought this extra dollar from the USDA, and we are now challenging ourselves basically to expand our welfare and our behavior-based measurements to understand like how the birds are playing with the laser and how that affects overall performance. So we found we had some initial questions just from people that had read our original work. And they were wondering, well, how many birds in the pen are actually following and playing with the laser, and do the birds get bored with that laser over time? So we've got two different sets of data here. One is days 0 to 8, and then it's also shown to you on the right, panel B, by week. And we saw that birds played with the laser in the first week on the first and the second days the most, because it was the most novel, and that kind of reduced down to day 8. And then if we look over time, there is typically a very small percent that are actually interacting with the laser. But we weren't too concerned about that, because we found that anywhere from 20 to 60% of the pens with laser enrichment were moving because of those birds that were interacting with the laser. So we were influencing a lot of movement in the pens just because a few birds were interacting with the laser. So we thought that was pretty interesting. And one aspect of our work in the smaller pens is that the birds don't have as many places to go rest. So we were kind of interested to know if they wanted to rest, could they still rest? And the answer was yes. Like, not everybody was running around like a madman because one crazy bird was very, very into that laser. So we thought that was pretty cool, and also important because we don't want to, force exercise. That's not our goal. But we found that even through week 6, birds were still like interested in getting up and moving. Maybe less so when they were chicks, but they were, still doing more activity. So in early this year, we completed our study where we lengthened the laser periods, and we were interested to know if the birds still kept moving over the 6-minute periods instead of the 4. And the answer is yes, they still do continue to move with the increased laser periods. The total percent moving based on day of life, so on the top left is 0 to 8, and on the bottom right is the weekly averages, but they still do move about the same as the birds with the shorter laser periods. And then my graduate student Megan and just a ton of undergrads did some really excellent work watching video, and they looked at each minute of the laser period, and they looked at the total percent of the birds moving. And basically we found that the birds don't really seem to get bored with the laser even through minute 6 of it being on. So this is through the first 8 days of life, and then this is through the first 7 weeks on a weekly basis. We thought that was kind of interesting, because we thought, they'll be bored with it at some point, but they didn't seem to be in this case. Then we also had a question early on was, are the birds stressed by using the laser or seeing the laser? And what we found was that by drawing blood from birds week 5, 6, and 7, we found that the serum corticosterone was reduced in the birds that had the laser enrichment. there seems to be a bump at week 6, and we're not really sure why the overall cohort would be higher, but we're still trying to kind of figure out the nuances of that. And again, I know serum cohort is not the most perfect measurement, but we were just kind of interested in it as a measurement, and we did find a reduction based on laser enrichment. So if you kind of take the performance and the feed conversion, the fact that they're choosing to interact with the laser or not, and then the serum cohort, I think we are kind of on the path of this is not a stressful thing for the birds. They can choose to play with it. The performance is telling you they're still eating and drinking equally to birds that are not laser enriched, and they're from the serum cohort measurement, they're not stressed out by exposure to the laser enrichment. So where we're kind of going right now is the question of is like, how can we measure if birds think woody breast is painful? And we're kind of at the beginning of thinking about this because over the last 2 studies, we've seen a definite change in woody breast score distribution and the overall like normal, moderate, or severe based on whether or not they have laser enrichment. And in most cases, we see less severe woody breast scores because of the laser and more that are into the normal or moderate category at both day 42 and day 49. So we think that is just a really interesting outcome. And we're wondering if it has to do just with, you know, the extra movement and how the muscle itself is developing or, you know, keeping blood flow over time. So we're at the beginning of trying to understand, is woody breast painful? That's one of our questions that we would really like to answer. We were also interested, so the last study, we did more measurements on the breast fillets. We were wondering if they weighed more or less. So we found that at day 42, they weighed significantly more. They were equal at day 49. But at day 49, we found that the breasts were taller in the laser enriched birds. And then at day 42, we found that they were wider. So we were wondering, how is our exercise changing some of the muscle physiology? Because we don't want to like have a global change to the product in the end. But so these are some of the changes that we found based on the characteristics. So I'd like to thank all the students that are behind the data. Kristen, Megan, and Kayla, they're grad students of mine. They're awesome, and we have just a ton of undergrads that do the work. But we went to San Antonio this summer, and we also went to Paris. So that was a very fun trip for the World's Poultry Congress. So had a chance to hang out with the grad students there. And then, of course, our great collaborators, Anna Johnson, Yuko Sato, Mo Elgazar, Billy and Danielle, amazing. And then our grant sources. So thank you so much for your time today.
All right. Do we have any questions from the audience? Or on Zoom?
Thank you for your presentation. I had a question about your seahorse assay. I know that you were looking at total PBMCs from the blood, but I was wondering if it was possible, or if you've even done it, to look at the glycolysis metabolism in different subpopulations of T cells or anything like that, if you've seen differences.
Yeah. So one of the changes that we see with vaccination or an immune stimulus is a total global change in who is around for the T cell populations. And our goal over the past year is we've been using a couple of different magnet kits to try to isolate out just T cell populations. The kits have been annoying and frustrating because we have to kind of use a sandwich technique. So we're working on making sure the cells that we get are the cells that we expect. And then we want to do the seahorse assay exactly as you said in those populations, because I think it would be different. We do have some information that we might know a couple of cell types that are driving those changes, but we want to be able to apply the challenge like just to that cell population or take it from a challenged animal. And then, yeah, so that is great. That's exactly where we're going. We're just working out the kits, as you know.
You mentioned that when birds were brought into a facility, 3 days of acclimation were not sufficient. Can you recommend what would be a reasonable amount of acclimation before you would begin an experiment?
Yeah, so the ICOOK minimum is 3, but 7 would, from an immunological standpoint, anything after 7 or after would be perfect. So it's just a little bit longer, but not too much longer. So I would recommend 7 would be a smart thing, but fully understanding that, like when we bring day-old chicks in, they get put on their feed right away. So yeah, within the bounds of your experiment.
I just would like to request that our virtual attendees do, if they have a question, to post it in the chat to everyone. I'm not going to pull up the chat on the screen. That's a direct message.
I was just wondering, your seahorse experiments suggest, though, that one could easily follow this same metric if one looked at small metabolites in plasma.
Yes.
On a mass spec or something.
Yeah. Yep. So you could definitely look at glucose or specific amino acid metabolism. Yeah.
Good morning. Thank you for your presentation,
Hi.
Dr. Bobeck. Is there a correlation between the laser enrichment and the heterophil lymphocyte ratio as an indicator for elevated levels of corticosteroid?
So we haven't done the H/L ratio in these particular birds, but is definitely something that we could add. We also haven't done our typical immune experiments because we've kind of been, I guess, developing where we want to go as far as sticking towards or moving towards a little bit more meat quality and stress, but I'm interested in doing H/L ratio. I'm also interested in drawing some blood and just looking at overall cell populations. And I'm also interested in adding maybe a secretory IgA in feces. So those would all be extra indicators. So we did the court and we just didn't know. Like, oh, interesting, because those results can be unreadable, right? But we had very tight standard errors and it just kept happening week after week. So now that we trust, there might be something we're willing to add more assays instead of doing the bomb approach and then finding out nothing worked. So yeah, thank you.
We have some questions on the chat here that I'm going to ask, and I have an anecdote that's very impressive that you have tight standard errors with your court measures, because usually CVs are insane. So that's great. Glad you get those. So we have a question from Billy Hargis here. Have you considered introducing the lasers after the first week?
Yes, yeah, we definitely have. So we've been able to take the lasers into a commercial facility, and something that is noticeable that is maybe less so in our research level is the lights are brighter in the first week and the birds are interacting with the lasers, but they don't discern them as well just because of the difference between the light and the red color of the laser. So we have thought, what if we let the birds, settle in for 10 to 14 days and then add it after that? Our initial trial where we just took the lasers in to see what birds would do with them, the birds were in the middle to the end of the grower phase, and they started playing with the lasers immediately. So I think the novel object would still apply even if they got the enrichment later. So definitely yes.
Okay, that is interesting. So speaking of play behavior, we have a question from Ruth Newberry asking, does the laser move at a variable speed in random pattern?
Yes, yeah. So when we were trying to decide what speed to put the laser at, we calculated how fast a fly moves. So we've had some difficulties, I would say, with our new laser system just because of what the motor is able to do, but we have it on a very slow setting, so it moves and it pauses, and what we're finding the birds are doing is they're pecking at it and then they're looking to see where it went, and as long as it moves within a short distance around them, they then run to that spot and peck and play with it. And it's definitely kind of in a randomized pattern because that's more interesting to them. But we've also found if we slowly move them towards the feeders, they end up camping out there. So it can be a good thing to move birds around, but One thing we don't do with it is we don't put it on the walls because that scares them quite a bit. So we keep it towards the middle of the pen in a concentrated area. And so birds that don't want to interact with it don't have to, and the ones that want to have that like play space. So yeah.
That's really interesting. So only on the ground, the laser's only on the ground, and if it hits the wall, they're scared?
Yeah, so it because of the way the laser is built, it also speeds up as it gets farther out in its arc. And many of the producers that we've interacted with, at least, I can't speak to everyone, but many of them use infrared thermometer to move birds off the wall to pile. So the birds are kind of already conditioned that, oh, I have to get up and move, and they kind of run away and have that like, oh, you know, scream, if you will. So we don't, it's not our goal. Our goal is to initiate, you know, play or curiosity and not fear off the wall. So we've been keeping it in the middle of the facility, and then the birds have the choice. And then there are also birds who just love hanging out on the wall, and then it doesn't disturb those because we don't want to pile any birds.
Always got to have those individuals
The
that
wildflowers.
don't do the same.
Yeah.
All right, we have Casey Guthrie would like to know what color of the laser that was used, and have you tried different colors, red versus blue, etc.?
We selected red because it is a tasty color. But also, as I've been learning, the way that the colors are made, we weren't initially interested in trying different colors, but the way that the lasers are made also dictates their intensity. So a green laser could blind a human or a bird. So our goal is to not do that. And we're staying in the red range because blood, right? It's a great color that the birds have always preferred to interact with. But for human and bird safety, red is not going to blind a bird or a human, but the green could. So we're not that. That's not our goal.
Any other questions from the audience? Or speakers?
Yeah, very interesting. I've had done some work with lasers too, and I do see an interest. I was wondering, and I was just thinking about it listening to your talk. With cats, I know they recommend going to another toy after laser play because it could cause some frustration. Have you seen any signs in chickens that they could respond the same way?
So I think the interesting thing that we found is we always track like single colors of birds when we color them on their back, so we know that it's a focal bird and we kind of have an idea of what they're doing as far as behavior. One thing we haven't tracked is repeat or preference for a laser. I'm kind of interested in that to know if that specific bird always likes to chase it, because we do see differences in the commercial facility. Like there seem to be birds that are really interested in it, and those that just don't really want to interact with it. But something that Megan introduced in this last trial is a novel object introduction. She's still working on that data, but we were kind of interested to see if we put another novel object, are they more likely to go and play and interact with that or not, based on whether or not they've had the laser. So we're still waiting on that data. We generate like thousands of hours of video, so that's why we've got the gaggle of undergrads. But yeah, we're interested to know if we know that they're not really getting tired with the laser over time, but they are growing bigger, so there's a increased preference to lay down anyway. But we don't want to introduce fear, so I think having a play area and a not play is important no matter where it goes.
So you're saying that you only run the laser in certain areas of the pen?
Yeah.
So they know to expect it there maybe? Or...
Yeah, so in our research, we have less of an ability to kind of control. At the commercial level, we have areas where the laser goes and where it doesn't. So you see birds kind of coming from the outskirts and running to go play with it. And then you see birds that are running from the bird running because they don't know why it's running. And then you see birds that are like, I'm out, and they just, walk away and do something else. So there's a lot of interesting responses, probably based on pecking order and some other, other things in the birds. But the commercial, it's just because of the size of the birds. Like in our pens with less than 40 birds, we have not seen huge piling behaviors, even with their not ability to go elsewhere based on where that laser goes. But commercially, we absolutely do not want piling behavior if there's something that is perceived as scary or a threat. So that's why we want to have the play area and then I don't want to do that. So that is something that we discovered as we moved into the commercial buildings. That's been really important and also kind of fun to watch.
Well, I'm gonna figure out some Zoom controls here, so why don't we take a space here?
Thank you.